Let me share my screen here. Can somebody confirm you can hear me?
Yes, we can hear you.
Thank you. All right, today is October 17th, and we're supposed to be covering Chapter 3, Observing Microorganisms, and hopefully we'll finish Chapter 3 today. Please note you will have a quiz Wednesday, Quiz 2. It'll be over Lab 3 and Lab 4. Excuse me, as well as Chapter 3 and the end of Chapter 2. Let me just check to make sure that quiz is up and running. I don't have that here yet. Come on, get out of the way. Oh, there it is. No? Slow today. Points. It looks like I might need to do something here. Let me look here. Oh, there it is. Quiz 2C. Yeah, that's not highlighted. Many of you guys can't see that. All right. So Quiz 2C is up and running. It'll be worth 75 points for all the quizzes. Yeah. All right. So it's up and running. That's good. I just wanted to double check. There was a problem with quiz 1, and that is there was a quiz 1B and a quiz 1C. Quiz 1C was the quiz you were supposed to take. Quiz 1B is actually, I moved it way down here because it's not a quiz I want you to take, and somebody got in there and took the question, so I can't delete it. So I'm going to make this one worth 0 points. I haven't done that yet, but I will do that. So this will not count towards the grade. And the person who took the quiz says they want to take quiz 1C anyway, so they'll get the grade for that. All right, any questions about anything before we begin? There is going to be a lab today. Lab 5, Streaking for Isolation and Culture Media. Let me remind you, you should be working on your infectious disease project. If you have not cleared a project with me, you need to do that before you begin. There are a few people I've told, I don't want you working on this project because it's too broad. You have to narrow it down. One way to narrow it down is like Staphylococcus aureus causes many diseases, and if you narrow it down to like blood diseases or bloodborne diseases or skin infections, I'll accept that. But I don't want you to do a project on a very broad topic because you'll have a hard time talking about it. And the best way to do it is if something like pneumonia, is you just pick one organism that causes pneumonia and then do your report on something like that. There are other ways to break it down, but That's an easy way to do it. Any questions
Is,
about any of that?
is there a page requirement for the paper?
Yes. I don't remember off the top of my head, so let me look that up. I think it's 5 pages. Oops, that's infectious disease project. All right. Finding it directly, but if I remember right, it's 5 pages. If you have uh, less than 4 pages, you'll get a penalty. If you have more than 6 pages, you'll get a penalty. This counts only the text. It does not count any figures or the references. Let me see if I can find that.
Okay, that's fine. You don't need to continue
There it is
looking.
right there.
That
Does not
helps. Okay,
exceed 5 single-sided pages right there.
but no less than 4, correct?
No less than 4 pages, yes.
Okay, thank you.
And that's including the text, not the references or the pictures. All right. Hmm. So last time we talked about the gram staining, and I think I went over this slide where I mentioned that first we stain with crystal violet and all the cells stain bluish purple. Then we add iodine. Iodine is a mordant. It intensifies the stain, the crystal violet, and iodine actually complexes with crystal violet, making a larger complex, which is why it intensifies the stain, and then it is harder to leave the gram-positive cells. We then do the decolorizing step, and it pulls the crystal violet iodine out of the gram-negative cells, but not out of the gram-positive cells. So this is the differential step of the gram stain. And then we stain with safranin, which stains the gram-negative cells pinkish. So the gram-positive cells are stained purple. The gram-negative cells are stained pinkish. Any question about that? So let's go through the gram stain in greater detail. Like I said, whenever you're doing the gram stain, you do it on a bacterial smear. So you have to make the bacterial smear first, and then you stain that. The bacterial smear is not part of the staining, and then the staining is not making a bacterial smear. Although to do the gram stain, you have to make a bacterial smear. Once you have the bacterial smear made, you then air dry it and heat fix it. Once again, that is not part of the gram stain. You then begin the gram stain. You first flood the slide with crystal violet, just covering the entire glass portion of the slide with crystal violet, and you let it sit for 1 minute. You then dump off the crystal violet and rinse it in cold water, rinse it gently. A crystal violet sticks to the peptidoglycan in the bacterial cell wall. It's considered the primary stain since all cells will pick up the crystal violet's color. Any question about that? As I stated, you wash the slide, and after that step, the gram-positive cells will stain purple, and the gram-negative cells will stain purple. You then, obviously, you dump off the worst of the water. You don't need to dump off all the water, but dump off the worst of the water. You then flood the slide with iodine. Iodine is a mordant. It increases the affinity of the crystal violet or the specimen, making a larger complex. Once again, you treat the slide with iodine for 1 minute. You then dump the iodine off and then gently wash it with cold water. After the iodine step, the gram-positive cells are purple. It is a more purple color than the crystal violet, which was a blue purple. But the gram-negative cells are the same color, and they're a purple color. Any question about any of that? You then have the decolorizing step. There are different decolorizing agents. We're going to talk about acetone ethanol as the decolorizing agent. It washes the crystal violet out of the gram-negative cell wall, but not out of the gram-positive cell wall, as long as you do not overwash. And I'll talk more about that in a minute. This is considered the differential step in the gram stain because the gram-positive cells will stain different than the gram-negative cells. What you do is you just apply the decolorizing agent for about 5 seconds. And this does depend a little bit on what your decolorizing agent is. We're going to talk about acetone ethanol as the decolorizing agent, but it could be acid ethanol, and then it could simply be ethanol. Those are the three. You don't need to know this, but those are the 3 decolorizing agents. If you use acetone ethanol or acid ethanol, you just apply the decolorizing agent for 5 seconds, sorry. What you do, the best way to do it is to actually observe the slide. Hopefully you can see this. I'm pretending this is my slide. And then you add the decolorizing agent across the top of the slide, watching it run down. Once the blue or purple stops running, you stop adding the decolorizing agent. And this will be about 5 seconds. Any question about that? So that's the best way to do it. If you stop and it's still running blue, you won't remove the crystal violet from the gram-negative cells. And if you continue doing it after the blue stops running, you will actually start pulling out the crystal violet from the gram-positive cells. So it is critical, this decolorizing step. You then wash the slide immediately with cold water. Why you do it immediately is after you apply the decolorizing step, there are trace amounts of decolorizer still on the slide. Those trace amounts will pull out the crystal violet from the gram-positive cells if it is left on. So if you notice, in all the other washing steps, I don't say wash immediately because it's not important. You could wash even 10 minutes later, assuming the slide doesn't dry out, and it would be fine. Here, you need to wash immediately to get the decolorizing agent off of the cells. Any question about any of that? At this step, the gram-positive cells will stain purple from the crystal violet iodine complex, and the gram-negative cells will be colorless because the decolorizing agent will have pulled out the crystal violet iodine from these cells. You then apply safranin. Safranin is a pink dye. It does stain all of the bacteria cells. We call it the counter stain because it has a different color to the primary stain, meaning it's a different color than crystal violet. You just flood the slide with safranin for 1 to 2 minutes. And you'll note that with my procedure, if you say, oh, I just flood the slide all the time with 1 minute, except for the decolorizing step, then the procedure is simple to remember and carry out. After 1 to 2 minutes, you then dump the safranin off and then wash it gently with cold water. Any questions about any of that? All right, after applying the safranin, the gram-positive cells will be purple color from the crystal violet iodine complex staying in the cells, and the gram-negative cells will be pink from the safranin. I already mentioned you wash that off with cold water gently. You then blot the slide with bibulus paper. Bibulus paper is a very thick, porous paper, much thicker than normal paper. It's kind of like a gentle, thin cardboard, but you'll probably never be in the lab, so you'll never probably get to see bibulus paper. Anyways, pretend this is the slide. You rip off the bibulus paper, and then I like to fold it. You don't have to fold it, but I like to fold it. And then stick the slide in between the bibulus paper and then blot it dry. That means press gently on the bibulus paper. You do not rub the slide up against the bibulus paper or the bibulus paper up against the slide, because that will rub off the cells. Although you could rub the bottom, but the top, you do not rub. You just blot it dry. That gets away enough water so that then you can observe the slide under the microscope, even if you use oil. And usually you will not start with 100x under the microscope, but you then move up to 100x to look at the slide, to look at the cells. Any question about any of that? All right. So the gram-positive cells are purple after the procedure of the gram stain, and the gram-negative cells are pink. Why? What's the basis of the differential staining? It's related to the differences in the cell wall. The gram-positive cells have a different cell wall than the gram-negative cells. The gram-positive bacteria have a thick peptidoglycan layer. Peptidoglycan is a disaccharide with amino acids. We'll talk a little bit more about that in a later lesson. Anyways, the crystal violet iodine complex gets stuck in the thicker peptidoglycan layer of the gram-positive cells. The gram-negative cells do have a peptidoglycan layer, but it's much thinner. And because it's thin, it cannot retain the crystal violet iodine complex. And so it washes out in the decolorizing step. Any question about any of that? So let's look at the basis of the gram stain. Let me blow this up. So here we see our cell. There's a gram-positive cell. It has a fairly thin yellow layer, which is the cell membrane, plasma membrane, and then a thick peptidoglycan layer of purple. This is many layers thick. This slide is only showing you 3 layers of peptidoglycan, but at a minimum, it's about 20 layers of peptidoglycan in the gram-positive cells. And it could be, depending on the cell, it could be hundreds of layers of peptidoglycan, but a minimum of 20. Compare that to the gram-negative cells, and we see there is a thin purple layer in between 2 yellow layers. The yellow layers are lipid membranes, the bottom one being the cell membrane once again. My mouse died, there it is. And above the peptidoglycan, there is another lipid bilayer shown here, and we're going to call that the outer membrane of the cell wall. So it is a part of the cell wall, but it's not peptidoglycan. It is a lipid bilayer. Any question about that? And then there is a peptidoglycan layer. This is only showing you it 1 layer thick, but in reality, the peptidoglycan has several layers, a minimum of 3 and up to about 20 layers of peptidoglycan in the gram-negative cell. All right, any questions about that? Oops, too far. So the crystal violet iodine complex forms in all cells, both the gram-positive and the gram-negative, after the addition of the iodine. The crystal violet iodine complex is larger than crystal violet or iodine alone. And during the alcohol acetone wash, the decolorizing step, in the gram-negative cells, the crystal violet iodine is washed out of the cells because the peptidoglycan layer is thin. And then the lipopolysaccharide layer, that's the outer membrane of the cell wall, is disrupted by the alcohol wash. And so the crystal violet iodine washes out. The cells will therefore be colorless after the decolorizing step. In the gram-positive cells, the crystal violet iodine complex cannot wash out of the intact, thick peptidoglycan layer. The cells therefore remain purple. Here we're looking at some cells stained with gram stain. You can see gram-positive cocci here, gram-positive rods or bacilli stained here, gram-positive coccus stain there, a gram-negative spirilla there, and then a gram-negative rod there. And that's either a vibrio or 2 rods connected together. That looks like a gram-negative rod. Any question about any of this? All right, in the gram stain, there are some difficult situations. Like, what do you do in this case here? What do you count these? Are these gram-negative and these gram-positive? What you do is you take a look at the stain and you base your color on the good regions of the slide. Regions where the cells are fairly a single layer thick. They're not clumped together. And then you see this, come on mouse, this dark blue there and this dark blue there and that dark blue there. That's not cells. What those are crystals of crystal violet, where the crystal violet has gone out of solution and actually formed crystals. And then when you put it on the slide, sometimes the crystals will come on the slide. And you'll note that the cells in this region, as well as this region, are darker than the cells further away. And so these look bluish purple, and those are more of a pink purple. And what you do is you count the, or determine the color away from these crystal violet crystals, because they can color the cells nearby. Any question about any of that? So make your determination of the gram stain in the good regions of the cell slide. Most consistent results are obtained in the gram stain when it's performed on young growing bacteria. If the bacteria are older than 2 days old, you should not use those cells to stain the gram stain. What you should do is grow the cells up and then use cells that are 2 days old or less to do the gram stain. The gram stain is not universally applicable. Some bacterial cells stain poorly, if at all, and that's mostly the mycobacteria, which stain poorly or not at all. So the results are not always as clear-cut as the theory suggests, and this may become evident during your unknown project, where you'll be pulling your hair out because the gram stain didn't quite work the way it should have. If you were actually doing the gram stain in the lab, you would learn that very easily, that the gram stain is a little tricky. It takes a little bit of practice to get it down right. Any questions about any of that? And it is a little tricky. All right. The structure of the gram-positive and gram-negative cell walls not only affects their staining, it also is related to how the cells respond to certain antibiotics. For example, gram-positive bacteria tend to be killed easily by penicillin G and some other antibiotics like the penicillin V and the cephalosporins, as well as many detergents. And that's because these, what do you want to call it, antibiotics or detergents, can get through the cell and do damage to the cell. Whereas the gram-negative outer membrane can prevent these antibiotics from getting to the cell, and they can prevent certain detergents from getting into the cell and then doing damage to the cell. So gram-negative bacteria are more resistant to at least these antimicrobials, penicillin G, penicillin V, the cephalosporin. And that's simply because they cannot get into the cell through the outer membrane of the cell wall of the gram-negative cell. And that's the lipopolysaccharide layer. Any question about that? Does that mean the gram-negative cells are harder to kill with antibiotics than gram-positive cells? Not really, because there are different antibiotics. Some of them work on gram-positive cells and not on gram-negative. Penicillin G's the only one we're going to really talk about that way, but I already mentioned penicillin V. And then there are other antibiotics, which we're not going to really talk about, so you don't need to know this, that work only on gram-negative cells. Oh, I had it in the top of my tongue, and now I've forgotten the antibiotic. Ah, gosh. Damages the cell membrane. It'll come to me. Starts with a P. Has a B in it. I'll have to think about that. There is an antibiotic that only works on gram-negative cells. It does not kill gram-positive cells. And then there are other members that kill both gram-negative and gram-positive. ampicillin, one of the early members of the penicillin family, actually works on both gram-negative and gram-positive. Most members of the penicillin family that were developed later, like carboxycillin, work on both gram-positive and gram-negative. Any question about any of that? Let me see if I can remember which one I'm doing. There's even a cephalosporin that only works on gram negatives, but that's not the one I was trying to think. Polymyxin, there we go. I remembered it began with a P. Polymyxin B is the antibiotic I was trying to think of, and it only works on gram-negative cells. Any question about that? So we do have antibiotics that do work on gram-negative cells. So later on, don't tell me that gram-negative cells are harder to work on to kill because penicillin G doesn't kill them. There are other antibiotics that we have that work on gram-negative cells, including ampicillin, which is a member of the penicillin family. All right, any questions about the gram stain? Here's a question for you. See, nobody answered a question, so I'm going to ask a question. I saw it in here someplace, further back. Here we go. We said that safranin stains all the bacteria cells, and that's why the gram-negative cells stain pink. Why don't the gram-positive cells stain pink after we stain the cells with safranin? I just stated right there, safranin stains all the cells. Or why isn't it a pink purple color after staining with safranin? Anyone have a guess?
Is it because the cell wall is thicker on the gram positive
Positive?
cells, and so it already stuck?
It's what? It's already
Is
stuck?
it because the cell wall is so much thicker on the gram positive?
It does have something to do with that, but I'm not understanding why the gram positives aren't staining pink just because the cell wall is thicker. Are you saying the pink doesn't get in the cell, or it's not staining the cell, or what?
I don't know. I was just speaking up.
Okay. All right. Actually, if you look at the gram-positive cells with an electron microscope, you can see, so let's pretend this is a cell here. You can see in different regions of the cell that there will be crystal violet iodine color in certain regions of the cell, and there will be safranin in certain regions of the cell. But to see that pink, you have to have the electron microscope magnification. Okay? When our eyes look at the cells, because the purple is a more intense color, all we see is the purple. Our eyes do not see the pink. Just the way our eyes operate. The purple is the more intense color, so our eyes only see the purple. The purple kind of hides the pink. But if we were to blow up the cell with an electron microscope, we would see that certain regions of the cell do have the pink dye in it, and other regions have the purple dye in it. So the pink dye is there, our eye just cannot see it. because it's hidden by the purple of the crystal violet iodine complex. Any question about that? All right, no questions? I think that's it for chapter 3. I want to get started just briefly on chapter 4, but it looks like we're going to have a short lecture today. I'm sure you guys won't complain. Any complaints?
No complaints.
So I just want to get started on chapter 4. Oh, wait a minute. I'm not finished with chapter 3. That's right. I got 2 more slides to talk about on chapter 3. And those are special stains. You're not going to be examined on special stains, but you may need to do a special stain for your unknown project. So I briefly want to talk about special stains. These are stains that we use for staining special properties or structures of the cell. One special stain that I want to talk about is the capsule stain. We use it to stain for capsules around the cells. Some of you may need to use this stain during your unknown project. The capsule stain is also called the negative stain, which is maybe more apparent in this one here. Because it reminds people of the negative in a Polaroid film, if you're familiar with that. In the old days, they used to print out the film for you, and then they gave you a negative. The negative would have the light areas dark, and the dark area is light, so that's why they called it a negative. And the negative stain reminds people of that. In the negative stain, the capsule stain, the background is colored. If the India ink capsule stain, the background is very, very dark. And then it stains the cells lightly. So the cells are light grayish color. Let me blow that up. And then the capsule is unstained. So it's very easy to see the capsules here in the capsule stain. There we go previously. There are other capsule stains. This one is using probably 2 different dyes. One to stain the background, a light purple color, and then a second stain to stain the cells, a dark purple color. Once again, the capsule does not stain. It appears clear. And you'll notice that in some places, it looks like the capsule has washed off, like right here, washed off its cell. But we can still see it. Probably what happened is like there's 2 there and there's only 1 around it. Probably one of them lost it. It might be that one there. Any question about any of that? Anyways, the capsule stain allows you to see the capsule because it doesn't stain with the capsule stain. And the background and the cells do stain. Any question about any of that? All right, that's the special stains. Let's finish chapter 3 and begin chapter 4. Chapter 4, the functional anatomy of prokaryote and eukaryotic cells. We will talk about the anatomy of prokaryotic cells. If we have time, I'll talk about the anatomy of eukaryotic cells. However, you were supposed to have eukaryotic cells in your previous biology class. I don't remember. Is that Biology 160? Anyone know at Clark College? I think it's
Yeah,
Biology
I
160.
think so, yeah.
So you should have had that. So it should only be a review. If we do not have time, I will skip the anatomy of eukaryotic cells, but we will cover the anatomy of prokaryotic cells. In this slide, I've got the major goals and the rough outline stated. So know the terms. What's the coccus? What's the bacillus? What's the spirilla? I think we've already talked about those, but we'll talk about it more. What is a strepto, like a streptococcus, a staphylococcus, a diplococcus? What's a tetrad? What's a Sarcenia? What's a vibrio? What's a spirocate? What's a glycocalyx? What are flagella? What's an axial filament? What are fimbriae? Fimbriae. I thought that was fimbriae. I'll have to look that up. And what are pili? What's peptidoglycan, endotoxin, and selective toxicity, selective permeability, sorry. Diffusion, facilitated diffusion, osmosis, active transport, tonicity, plasmid, inclusions, what are endospores, and then lastly, we will talk about the endosymbiotic theory. The endosymbiotic theory is actually the anatomy of eukaryotic cells. So it is fimbriae. I've been saying it wrong. Fimbriae. So that's the only part of the eukaryotic cells that I promised we will talk about. Regardless if we have time or not, I will cover the endosymbiotic theory. So we'll end with that. In this overview, also know too how gram-positive and gram-negative cell walls differ. So we'll talk about that in a little more greater detail. And then 3, describe the importance of taxus in bacteria. Any question about any of that? All right. When we're talking about prokaryote cells, comparing them to eukaryotes, you should know that prokaryote is the Greek words for pre-nucleus. Literally, it translates to pre-kernel, but by kernel, they're talking about nucleus. And eukaryote is the Greek word for true nucleus, or literally true kernel. Prokaryotes generally have one circular chromosome. That means the chromosome is in a circle, and the beginning point of the chromosome is the same as the ending point. The chromosomes are not in a nucleus in a prokaryote. In the eukaryotes, the chromosomes are generally linear, like on the X chromosome, shown by my X here. It has a beginning point and an ending point, and those are 2 different points on the chromosome. It's not circular, and the chromosomes are in a nucleus. For eukaryotes, the DNA is not associated with histones. Histone is a protein, and it's the major protein on the eukaryote chromosome. So in eukaryotes, our chromosome has both DNA and histone proteins. In prokaryotes, the chromosome does have DNA and it has other proteins, but it does not have histone proteins. If you really want to know, in eukaryotes, the histone protein is a very large protein. Maybe I can draw it. A very large protein, like that. And the DNA is this string that actually wraps around the histone. I'm going to have a hard time drawing that. And it actually wraps around the histone 2 times. And then this DNA is linked to another histone protein over here, which I didn't draw that one well. Let's make it like that. Okay. So that's how the DNA is held together in eukaryotes. Any question about that? There are no organelles in a prokaryote cell. By organelles, we mean membrane-bound organelles, such as the mitochondria, the chloroplast, the Golgi body. There are membrane-bound organelles in the eukaryote cell, such as the Golgi complex, the Golgi body, or the mitochondria, the chloroplast. And don't forget that one of the other membrane-bound organelles is actually the nucleus. It is a membrane-bound organelle. Usually I just use the word organelle when I mean membrane-bound organelle. There is peptidoglycan in the cell walls of bacteria. In fact, it's the main molecule in the cell wall of bacteria, if the cell has a cell wall. There is no peptidoglycan in the cell walls of eukaryotes. And of course, animal cells do not have a cell wall, so they don't have peptidoglycan either. Prokaryotes divide mostly by binary fission. That's where one cell grows and then splits down the middle in two. Eukaryote cells divide by mitosis. Any question about any of that? So in the chapter summary, we're going to talk about prokaryotes, talking about their cell shape and their cellular arrangement. We'll talk about different cell structures we find in prokaryote cells, and then we'll talk about the movement of material across the membranes. If we get to eukaryotes, we'll talk about the different cell structures you see in eukaryotes, but we may not cover this if we don't have time. If we do, I'll cover it, but usually I do not. And then lastly, we'll talk about the endosymbiotic theory, a theory for why eukaryotes have mitochondria and chloroplasts. Any question about any of that? All right. Prokaryote cell size and shapes. The average prokaryote is actually 1 to about 3 micrometers, the average. The very extreme range is something like 0.4 micrometers to around 10 micrometers long for the prokaryote. You can get some prokaryote cells which are smaller, but people believe that those cells are not viable. So about 0.4 is the smallest prokaryote that's at least viable. And about 10 micrometers is the longest. There are 1 or 2 prokaryotes which are actually larger. And I don't remember how large the largest prokaryote is. I think it's close to 100 micrometers. I should look that up, but there's very few that are that size. There might be 1 or 2. The basic prokaryote shapes are most prokaryotes are cocci, shown here. That is a round ball-like shape. The second are bacilli, a rod, shown here or there. And then the third is a spiral, shown here. There are other prokaryote shapes, but most of them are a derivative of one of these 3 main shapes. Like the Coccobacillus is a shape in between a cocci and a bacilli. If your bacillus has rounded edges, sort of like this one here, and then you shrink the bacillus, getting it smaller and smaller, pretty soon it'll start appearing to be a coccus. And those we call coccobacilli. Any question about the three main shapes? When we're talking about spirals, there are different types. This is the main spiral, the spirillum. You can have half a spiral, and you can call that a vibrio, which you can call that a spiral or half a spiral, or you can call it a bent rod. What a vibrio is, as far as a spiral or a bent rod, I guess it depends on who the author is. Actually, I call them a bent rod, but you can call it a vibrio, which is this type of a shape. And you can call it a half of a spiral. And then there's a spirochete. That's a spiral with many spirals in it. There are some differences between the spirillum, the spiral, and the spirochete. The spirochete is more flexible. The spirillum is more rigid. For example, the spiral does not usually, let me see if I can draw this, not draw it, but show you. The spiral does not usually come on itself, so we'll have to make this like a spiral, something like that. It does not usually go like that on itself. The spirochete can. Usually it's in this type of a shape, but it can bend on itself and be like that. And I'll actually show you a little video of a spirochete that did that during the video. Any question about that? There are some unusual shapes among the prokaryotes. We're not going to talk about them, but the Stella bacteria actually takes a star type shape. This is an actual electron microscopic image of the Stella, and there's an artist's rendition of it. And then the Haloarcula, it says square here, but if you look at that closely, it's more rectangular. And there's a Haloarcula right there. This is Archaea, and the Archaea do have a few odd bacteria. The Haloarcula is one, a bacteria that's actually what do you call that? Not a square, I just lost the name of this rectangle. Most prokaryotes are monomorphic, meaning that all members of the species have one shape. Like the Haloarcula, all of them are rectangles. The Stella, all of them are star-shaped. The Staphylococcus epidermidis, all of them are cocci. Okay? And we call them monomorphic. They have a single shape. But a few species are pleomorphic, like this species here. Let me blow this up. Pleomorphic means there are different types of shapes of cells. Usually the pleomorphic bacteria are bacteria that do not have a cell wall, and because they don't have a cell wall, they can take on different shapes. When we're talking about the oh, wait a minute, let me move forward here. For some reason, this slide has the Coccobacillus here, and it shouldn't be here because the Coccobacillus shown here is not a cellular growth arrangement. It is a cell shape where the cell is in between a cocci and a bacilli. And here we're looking at Coccobacillus, where they are a little bit longer than a cocci, but they aren't as long as a rod. They're not clearly a rod. And those we call Coccobacillus. All right, let's talk about the cellular arrangement, also called the cellular growth arrangement. These are when cells divide. Sometimes they stick together. With the cocci, you can have a cellular arrangement where the cells are singlets, a single cocci. In fact, let me blow that up. Right there, and right there, and right there, that's a singlet, a single cocci. You can also have diplococci. That's where two cocci are stuck together. And right there is a diplococci. Right there is another diplococci. You can have streptococci, where the cocci stick together in a row, a line, or a string. And there's a streptococci here. There's another streptococci underneath it. You can have tetrads. That's four cocci that stick together. And if you want to know, the reason why that happens is because the cells divide on two planes, both the X plane and the Y plane, creating the tetrad, whereas the streptococci, the diplococci, and the singlets divide on one plane. Why, when it divides, it always, in the streptococci, always goes this way and that way. If you get the cells dividing on the Y, X, and Z plane, you can get sarcenia, where there are 8 cells stuck together. Let me see if I can show you that. It's hard to see that here. Right there is a tetrad, 1, 2, 3, 4. There's 1, 2, 3, 4. The sarcenia, it's very hard to see because there's so many of them here, but there's a sarcenia. We're just going to go with that. And you can see everything except for that one cell right there. With the cocci, you can also have staphylococci, shown here or here. This reminds people of a cluster of grapes. You have the grape stem here, and there's the grapes. And staphylococci actually means cocci in the shape of a grape cluster. Any question about any of that? Anyways, those are all the cellular arrangements for cocci. Don't forget about the singlet. Students often forget that the singlet is a cellular growth arrangement or a cellular arrangement. With the bacilli, there are fewer cellular arrangements. You do have single bacillus or a singlet, and there's one there. There's another one there. You can have diplobacilli, which for me, I don't clearly see that here. There's one here and then one there, but there's the diplobacilli. That's 2 stuck together on the lengthways. So they're stuck together this way, which I suppose that doesn't matter too much for diplobacilli. There's streptobacilli, which are stuck together in a chain. So they are stuck together this way once again. And then there's palisade. That's where they're stuck together this way. Okay. If you ever read Mark Twain, I think it was Tom Sawyer was painting a palisade fence. And if you put this on its side here, it sort of looks like a fence made out of boards. And that's what a palisade fence was. A fence this way. It's supposed to be straight boards going into the ground below. And you can have bacilli growing this way. And it's called the palisade after the palisade fence. And then the ground is this on this side. Come on. Anyone remember the Palisade fence in Mark Twain? Nobody's saying anything, so I guess you don't remember and nobody wants to comment. I think that's it. Oh, I should note that when you're talking about bacillus, it does have two meanings. There is a capital B bacillus that is a family of bacteria. If it is a lowercase b bacillus, we mean the shape of a cell. Usually, actually all the time, almost, we'll say usually though, usually when you look at the cell arrangement of prokaryotes, they are usually a mixture. The diplobacilli can break, and so you'll see singlets as well as diplobacilli. The streptobacilli can break, so you can see diplobacilli, streptobacilli, diplobacilli, and singlets. And that's true of all the cellular arrangements. So usually it's a mixed bag, and then you simply call it by the highest order one. With the spirals and the spirochetes, they will only have singlets. So their cellular growth arrangement is only singlets. And they're the only cells where you only see, or you see all of the cells, one cellular arrangement, a singlet. For almost all the other cells, you will have mixed cellular arrangements. Any question about any of that? All right, if not, I'm going to stop here and then I'll see you at 6:30 for the lab. Any questions? All right, I'll see you at 6:30, assuming that's the correct time. Let me check on that. Yep, 6:30. Bye.